Learning outcomes. After studying this module, you will be able to know about biochemical genetics, about inherited genetic disorders, about various genetic disorders. Introduction. The diagnosis and management of inborn errors of metabolism is dealt in biochemical genetics. It deals with those patients which have enzymatic deficiencies that disturb the biochemical pathways involved in metabolism of lipids, carbohydrates and amino acids. If there is inherited abnormality in the biochemistry of body, then it is known as biochemical genetic disorders. Variations in these diseases is high and are incurable. Some of the diseases are related with mental retardation and other developmental issues. Some of them can even be life-threatening. Severe effects of such diseases can be prevented if they are detected and treated early. Early treatment can change the natural progression of the disease. Some of the metabolic disorders are glycogen storage diseases, lysosomal storage disorders, phenylketonuria, etc. Inherited metabolic or genetic disorders. These diseases result in metabolic disorders. In most of the cases, people with such disorders have a defective gene that results in an enzymatic deficiency. Hundreds of metabolic diseases are present with varied symptoms, treatments and prognosis. Metabolism. Chemical reactions takes place in body to convert energy from one form to another or to use the energy. These chemical reactions are referred as metabolism. Some major examples of metabolism are breaking down the carbohydrates, proteins and fats in food to release energy, transforming excess nitrogen into waste products excreted in urine, breaking down or converting chemicals into other substances and transporting them inside cells. Metabolism is an organized chemical assembly line, but it can also be chaotic. Production, transportation and excretion of raw materials, half-finished products and waste materials is constantly taking place. To carry out the chemical reaction, various enzymes and proteins are present. Causes of inherited metabolic disorders. The enzyme required for carrying out a particular metabolic process is either not produced or does not work even after production, results in inherited metabolic disorders. Due to inefficiency or absence of enzyme, building up of toxic chemical substances can take place and the main required products may not be produced. Pair of genes are required for production of a particular protein. In most of the cases, inherited genetic disorders, 2 defective copies of genes are inherited, one from each parent. In such cases, both the parents are carriers of bad gene, that is, each have one defective and one normal copy. Bad copy is compensated with normal gene in case of parents. Due to adequate enzyme levels, no signs of any such disorder can be observed. Since the child inherits both the defective genes, production of enzyme is inadequate, thus developing genetic metabolic disorder. This type of genetic transmission is called as autosomal recessive inheritance. The root cause of these genetic issues is gene mutation, which may have occurred many generations ago. This gene mutation passes along through the generations. Such inherited genetic disorders are not common in general population. These disorders can be seen in about 1 in 100 to 2500 newborns. The rate of genetic disorders is higher in certain ethnic populations such as Ashkenazi Jews, Jews of Central and Eastern European ancestry. Types of inherited metabolic disorders. Hundreds of inherited genetic disorders have been discovered. Identification of new ones is still continued. Some of the common genetic disorders include lysozyme storage disorder. Breakdown of waste products of metabolism take place in spaces present inside the cells known as lysosomes. Toxic substances are built up in lysosomes due to enzyme deficiencies. This enzyme deficiency causes metabolic disorders which include Hurler syndrome, abnormal bone structure and delay in development, Niemann-Pick disease. Babies develop liver enlargement and can have difficulty feeding. Nerve damage can also be seen. Tay-Sachs disease. Deletion or base insertion in chromosome 15 leads to Tay-Sachs disease. It is an autosomal genetic disorder characterized by loss of motor control, muscle strength and function, dementia and seizures. If defective copy of the gene is inherited from both the parents, only in that case the disease will be expressed. Production of hexosaminidase A is decreased due to this defective gene. Hexosaminidase A is a lysosomal enzyme that functions in the chemical degradation of gangliosides, which causes a built-in nerve cells in brain. Children suffering from this disorder usually die before the age of 5. Care for this disease is still unknown. Enzyme replacement therapy can be used in which the defective enzyme is replaced by a functioning enzyme. In addition, substrate reduction therapy, which treats the buildup of gangliosides by using other enzymes to catabolize it. A month-old child can show progressive weakness, progressing to severe nerve damage. The lifespan of child is very short. He or she usually lives only until age 4 or 5. Gaucher disease. Pain in bones with enlarged liver is one of the symptoms of this disease. Platelet count is low in children and adults. Fabry disease. Pain in the extremities is observed in childhood with kidney and heart disease and strokes in adults. Only males are affected with this disease. Krabbe disease. Delay in development in young children and progressive nerve damage is seen. Most often adults are affected. Galactosemia. Impaired breakdown of sugar galactose leads to jaundice, vomiting and liver enlargement after breast or formula feeding by a newborn. Maple syrup urine disease. Deficiency of an enzyme called BCKD causes buildup of amino acids in the body. Nerve damage can also be seen and the smell of urine is like syrup. Phenylketonuria, PKU. Mutation in gene coding enzyme phenylalanine hydroxylase, PAH, results in an autosomal genetic disorder known as phenylketonuria or PKU. This mutation makes the gene non-functional. Function of PAH is to metabolize amino acid phenylalanine into tyrosine. Building up of phenylalanine results due to absence of PAH activity. Levels of large neutral amino acids in the brain is decreased due to the presence of excess phenylalanine in blood, leading to hindered development of brain and severe effects such as mental retardation. Glycogen storage diseases. Problems with sugar storage lead to low blood sugar levels, muscle pain and weakness. Mitochondrial disorders. Problems inside mitochondria, which are powerhouses of the cells, can also lead to muscle damage. Friedreich ataxia. Problems related to a protein called frataxin cause nerve damage and often heart problems. Inability to walk usually results by young adulthood. Peroxisomal disorders. Just like lysosomes, Peroxisomes are also tiny spaces filled with enzymes present inside cells. Toxic products can also build up in peroxisomes due to poor enzymatic activity. Peroxisomal disorders include Zellweger syndrome. In this, abnormal facial features develop and nerve damage in infants. Enlarged liver is also one of the symptoms. Adrenoleukodystrophy. It is a disease in which nerve damage can develop in childhood or early adulthood depending on the form. Metal metabolism disorders. Special proteins control the level of metals in the blood. Genetic disorders can result in malfunction of proteins and accumulation of metals in the body, which can be toxic. Some examples of metal metabolism disorders are Wilson disease. Toxic levels of copper accumulate in liver, brain and other organs of the body. Hemochromatosis. Absorption of excessive iron by intestine causes damage to liver, joints, heart and pancreas. Organic acidemias. Methylmalonic acidemia and propionic acidemia. Urea cycle disorders. Deficiency of ornithine transcarbamylase and citrullinemia. Chromosomal abnormalities. In chromosomal abnormalities, mutation in chromosomes is present. Some of the chromosomal abnormalities are Duchenne muscular dystrophy. A rod-shaped protein is present in muscle and cardiac fibers known as dystrophin. This protein connects the fiber cytoskeleton to extracellular matrix of cell membrane. When mutation occurs in dystrophin, it results in Duchenne muscular dystrophy. It is passed from mother to the son. 2/3 of the causes are of inheriting this disease, whereas in 1/3 of the cases, new mutation of a specific gene can be seen. The mutation causes the alteration of the shape and function and a lack of production of the dystrophin protein, causing symptoms such as muscle weakness and heart problems like arrhythmias, cardiomyopathy and congestive heart failure. This is rarely seen disorder in about 1 in 3,500 boys. Most of them do not live past their early 20s. Females are not affected by this disease, but they are at high risk for developing heart issues. Angelman syndrome. Deletion or silencing of gene inherited from mother on chromosomes 15 causes genomic imprinting disorder known as Angelman syndrome. It is identical to another disorder which affects paternally inherited chromosome 15, Prader-Willi syndrome. Genes from both the parents are inherited, but due to specific deletion of a portion of chromosomes, the maternal genetic information is silenced. Angelman syndrome can also result from a single gene mutation on UBE3A gene. Seizure, mental retardation, jerky movements such as hand flapping, etc. are some of the symptoms observed. Support and therapy can improve motor skills and living as it is not a degenerative disease. Hemochromatosis type 1. Hemochromatosis type 1 causes overabsorption of dietary iron. Aggregation of excess iron can be seen in tissues in liver, heart, adrenal glands and pancreas. Accumulation of excess iron can result in cirrhosis, diabetes, heart failure and adrenal dysfunction. Mutation in HEP gene can result in this disease. HFE monitors and conducts the binding of transferrins, which are proteins that carry iron in blood. Concentration of these proteins is low in blood. Whenever low iron levels are exhibited, these proteins are released. which stimulate the secretion of iron in blood. Due to mutation of this gene, continuous signal is sent by intestine for release of these proteins, which leads to oversecretion of iron. Polycystic kidney disease. It is an autosomal dominant disease which is inherited from one parent. Polycystic kidney disease occurs in both adults and children. Cysts develop in kidneys, liver, pancreas, diverticulitis of the colon, and aortic and brain aneurysms. Huntington disease. It is a neurological disorder. It is an autosomal dominant disorder in which gene responsible for expression of protein Huntington is mutated. This protein is expressed in the brain. Repetition of glutamine residues takes place in Huntington. People affected by this mutation have the CAG array from 36 to 82 or longer repeats, while healthy individuals have an array between 6 to 31 repeats. The array becomes longer as it is passed from one generation to the next, a phenomena called as anticipation. Anticipation causes the children of an affected parent to have an earlier onset of the disease than the parent. Triple X syndrome. Females having more than 2 X chromosomes suffers from triple X syndrome. These females are usually tall and thin. Mental retardation can also be observed in some cases. Many phenotypic changes cannot be seen in females. Some can be infertile. Females with this disease can menstruate regularly and can have children. The severity of symptoms like mental retardation and other physical issues increase with more number of X chromosomes. Progeria. Progeria is a Greek word which means prematurely old. This disease is also known as Hutchinson-Gilford progeria syndrome. Jonathan Hutchinson and Hastings Gilford were the first ones to notice this disease. In this disease, premature aging of children can be seen. Babies seem to be healthy when they are born, but after few years they start displaying an unusual accelerated aging process and can lead to hair, aged skin, heart disease, stiff joints and osteoporosis. Most of the children die by their teens. Lamin A gene on chromosome 1. Turner syndrome. It is a rare genetic abnormality which affects the development in females only. Certain physical attributes can be seen in girls with Turner syndrome. The level of severity of this varies with individuals. Most of the cases of this disease are not inherited. It is characterized as a chromosomal abnormality when one normal X chromosome is present in a female cells and the other X chromosome is missing or structurally altered. Sickle cell disease. If there is RN hemoglobin gene, then it results in sickle cell disease. It is a recessive genetic disorder. Hemoglobin is the oxygen carrying protein in RBCs. The shape of blood cells is affected. It distorts into an abnormal crescent shape. Jamming of blood cells or their breakage can be seen due to distorted shape of cells. Severe pains in organs and joints is experienced by people suffering from this disease. This pain can last from hours to days. The blood vessel blockages prevent red blood cells from transferring oxygen to tissues and organs. Symptoms include abdominal pain, poor eyesight or blindness, ulcers and strokes. Summary. The diagnosis and management of inborn errors of metabolism is dealt in biochemical genetics. Biochemical genetics is also known as metabolic genetics. Variation in these diseases is high and are incurable. Hundreds of metabolic diseases are present with varied symptoms, treatments and prognosis. Chemical reactions takes place in body to convert energy from one form to another or to use the energy. These chemical reactions are referred to as metabolism. Metabolism is an organized chemical assembly line, but it can also be chaotic. The enzyme required for carrying out a particular metabolic process is either not produced or does not work even after production results in inherited metabolic disorders. The root cause of these genetic issues is gene mutation, which may have occurred many generations ago. Hundreds of inherited genetic disorders have been discovered. Identification of new ones is still continued. Toxic substances are built up in lysosomes due to enzyme deficiencies. Deletion or base insertion in chromosome 15 leads to Tay-Sachs disease. It is an autosomal genetic disorder characterized by loss of motor control, muscle strength and function, dementia and seizures. Mutation in gene coding enzyme phenylalanine hydroxylase PAH results in autosomal genetic disorder known as phenylketonuria PKU. Just like lysosomes, peroxisomes are also tiny spaces filled with enzymes present inside cells. Genetic disorders can result in malfunction of proteins and accumulation of metals in the body, which can be toxic. Absorption of excessive iron by intestine causes damage to liver, joints, heart and pancreas. When mutation occurs in dystrophin, it results in Duchenne muscular dystrophy. It is an X-linked recessive disorder. It is passed from mother to the son. Deletion or silencing of gene inherited from mother on chromosome 15 causes genomic imprinting disorder known as Angelman syndrome. Hematochromatosis type 1 causes overabsorption of dietary iron. Polycystic kidney disease occur in both adults and children. Repetition of glutamine residues takes place in Huntington. Females having more than 2 X chromosomes suffer from triplo X syndrome. Premature aging of children can be seen in progeria. The shape of the blood cells is affected in sickle cell disease. It distorts into an abnormal crescent shape.